Welcome to section 17.1, Viruses. In this section, we'll learn what viruses are, how they were discovered, how they reproduce, and how humans fight them with vaccines and antiviral drugs. Viruses are fascinating because they sit on the edge of life, not quite living, but not entirely inert either. They're incredibly small, incredibly simple, and yet capable of enormous impact, from common colds to global pandemics. By the end of this section, you should be able to explain how viruses were discovered and detected, describe the detailed steps of the viral replication cycle, and explain how vaccines help us prevent and treat viral diseases. Viruses don't leave fossils, so their discovery was recent compared to bacteria. In the late 1800s, researchers studying tobacco mosaic disease found that the infectious agent could pass through a Chamberlain-Pasteur filter, which blocked bacteria. That told scientists that they were dealing with something even smaller. In the 1940s, the invention of the electron microscope finally let us see viruses directly, including tobacco mosaic virus. This was like switching from a blurry magnifying glass to a high resolution camera for the first time. Viruses are acellular, meaning they are not made of cells. The complete infectious particle is called a virion. Each virion carries genetic material, either DNA or RNA, inside a protective protein coat called a capsid. Some also have a lipid envelope stolen from the host cell's membrane. You can think of it like a package. The genome is the instructions inside, the capsid is the box, and the envelope is like borrowed gift wrap. Viruses also come in many shapes, from rods to spheres, or even complex forms like bacteriophages that resemble lunar landers. Viruses are extremely small, measured in nanometers. A nanometer is 1 billionth of a meter. Most viruses range from 20 to 300 nanometers, while a typical human cell is 10 to 100 micrometers wide, 100 to 1,000 times bigger than a virus. If a human cell were the size of a baseball stadium, then a virus would be a marble on the field. Because viruses are so small, we can't see them with regular light microscopes. Visible light's wavelength is too long to resolve them. That's why scientists use electron microscopes, which use beams of electrons with shorter wavelengths to reveal virus structure in more detail. Viruses can't reproduce on their own. They need a host. The replication cycle has five main stages. Attachment is where viruses bind to specific receptors on the host cell. Entry, which is where the virus or its genetic material enters the cell. Uncoding, which is where the viral genome is released. Replication and assembly, which is where the host cell is hijacked to copy viral DNA or RNA and then build proteins. And then finally, release, where new viruses exit, either by bursting the cell or budding off gradually. The cycle shows why viruses are obligate parasites. They bring instructions, but they borrow the cell's machinery. Viruses are picky about their hosts. They can only infect cells with the right surface receptors. HIV, for example, attaches to CD4 receptors on immune cells. The SARS virus that causes COVID 19 binds to ACE2 receptors in the human respiratory tract. This lock and key system explains why viruses infect some species but not others, and why certain viruses target specific tissues inside one organism. Vaccines prepare the immune system by safely introducing it to part of a virus. Traditional vaccines use weakened or killed viruses. Newer vaccines use viral proteins or even genetic instructions like mRNA. Vaccines have eradicated smallpox and nearly eliminated polio. They remain our strongest tool against viral disease, even though some viruses, like influenza, mutate quickly, which requires regular updates to our vaccine strategy. Antiviral drugs don't kill viruses directly, but they disrupt key steps in the viral life cycle. Acyclovir, for example, blocks replication of herpes virus DNA. Tamiflu interferes with influenza's ability to leave infected cells. And HIV is treated with HAART, a combination of drugs that prevents the virus from replicating and mutating into resistant forms. Antivirals are harder to design than antibiotics, but they are essential for managing chronic viral infections. Viruses evolve rapidly, which means they can escape vaccines or become resistant to drugs. This makes ongoing research critical. Scientists are constantly working on new vaccines, new antivirals, and even exploring novel approaches like CRISPR based therapies. The challenge is huge, but so is the payoff. Our ability to respond to pandemics depends on innovation in virology. Viruses are unique infectious agents that blur the line between living and non living. They were discovered through clever filtration experiments and visualized with electron microscopy. Structurally, virions package genomes inside capsids, sometimes wrapped in an envelope. They reproduce only by hijacking host cells using a precise replication cycle. Host specificity comes from receptor binding. Vaccines and antivirals are our best defenses, but viruses evolve quickly, keeping scientists on their toes. Studying viruses gives us insight into disease and into some of biology's most fundamental processes.